Welcome. In this session, firstly we'll say about diagrammatic representation of rDNA technology. Then later we'll see separation and isolation of DNA fragments. And then later we'll also say about cloning vectors. So let's begin this session with the diagrammatic representation of rDNA technology. So in the last session, I told you that uh, after isolating, so we need to isolate the DNA, okay, from the source organism, and then we need to join this gene of interest. Okay, so this is the vector DNA. So that is the plasmid. So we need to join that the gene of interest to this vector DNA or the plasmid, and this is the foreign DNA. And I need to use the same type of restriction endonucleases in both the cases. Okay, so if I use a different restriction endonuclease to cut this DNA, foreign DNA, and to cut the vector DNA, then I will be not able to get the same type of sticky ends. So say suppose I'm using this echo R1 restriction endonucleases on both these cases. So then, okay, from this I'm going to get the gene of interest. So this is the gene of interest, and this gene of interest I need to put into this vector DNA. So here the vector DNA is also cut with the same restriction endonuclease, so hence it will have same type of sticky ends. And that later, once I join this, of course I'm going to take it into a test tube and then put this gene of interest and then this vector DNA together, and I'm going to apply something called DNA ligase. So that DNA ligase will help in joining this gene of interest with the vector DNA, that is plasmid. Okay, so this is with the help of enzyme called DNA ligase. Earlier, I used this enzyme, that is restriction endonuclease, right, to cut the foreign DNA and the vector DNA, the same restriction endonuclease. Then later, I used this DNA ligase to join this gene of interest, which I have taken, okay, from the source organism with the plasmid, right? And now I need to so that's what this becomes the rDNA. Now I need to put this rDNA into the bacterium. So this I'll be introducing into the bacterium. So this introduction into the bacterium is what we call it transformation. I'm just mentioning like this. Okay, this is the bacterial genome. Okay, say suppose this is the E. coli bacterium. Okay, and this is the rDNA that I have introduced into E. coli bacterium, and this is a process called transformation. I hope you remember from the chapter molecular basis of inheritance, so what we mean by transformation. Okay, so when this rDNA we have put into this bacterium, E. coli bacterium, I have transformed this E. coli bacterium, and now this E. coli bacterium is allowed to divide in a nutrient medium. So when it is allowed to divide into the nutrient medium, what happens is the plasmid which is present, or what I say, the rDNA which is present in the E. coli bacterium will also divide along with it. So that will also will divide, you can see, and thereby making the copies of rDNA. So that's what I call it gene cloning. Of course, that will take place along with the cloning of host. Right. So this is how we see in case of the rDNA technology, and I'm going to grow this E. coli bacteria or whatever in large number so that I can get the product in large quantity. Say suppose this gene of interest which I have introduced into this bacterium is producing a particular hormone, say suppose a growth hormone, and I want that hormone in large quantities. So I will allow this E. coli bacterium uh, to multiply in large number. So how we will do it, all that we will see in coming sessions. Okay, so now the first process of this is, you know how to get that gene of interest. So if I'm going to take human DNA, we know human DNA is a very long, okay, 2.2 meter long, and in that we need to find a specific region where the gene of interest lies. So of course there may be not only one gene of interest. In case of that, there may be several regions where we find the gene of interest. So one gene that codes for growth hormone, and one gene that codes for insulin, and another gene that codes for whatever. So we need to separate all these uh, genes from the DNA. Now for that, how we need to do it? So that's what I'm going to discuss under separation and isolation of DNA fragments. Separation and isolation of DNA fragments. Isolation of DNA fragments. Okay, and see, once I isolate, so that part I will discuss again later. So to remove this DNA from the cell, so we know that eukaryotic DNA will be present, okay, and it's complicated, present inside the nucleus. We have cell membranes, then nuclear membrane, and here it's the DNA, and that too, the DNA will be not in its purified form, means it is not just present as DNA. It will be associated with a lot of proteins, RNAs, and many other substances will be present. We need to remove all of them and make sure that it is only DNA. That's what I call purified DNA. Now, I will be using several enzymes. That part I will discuss okay later. I'll use several enzymes to remove this cell membrane, nuclear membranes, and the proteins, whatnot, all associated with the DNA. And I'm going to get a purified DNA. So once I get that purified DNA, what I'm going to do is take that purified DNA, okay, into a test tube, and then I'm going to add restriction endonuclease. So when I add restriction endonuclease, a particular restriction endonuclease, and this restriction endonuclease will cut the DNA wherever it finds that palindromic sequence. Okay, right, say suppose we'll take the same example, echo R1. So if I am taking this echo R1 restriction endonuclease, wherever it comes across this GAA-TTC, so this is the palindromic sequence that is identified by echo R1. So that may be present at different locations. So when this is present at different locations, then all that points it will cut the DNA, right? As a result, with this we will be getting the DNA fragments. I'm sorry. We'll get the DNA fragments. Okay, and those DNA fragments will be of various lengths. So some may be short, some may be long, with medium size and all. So these with various, what I say, lengths. So these are DNA fragments of various lengths we will obtain. So once I use that restriction endonuclease, now I need to separate these DNA fragments according to their size. Okay, and I will do it by using gel electrophoresis. So these DNA fragments I'm going to separate by gel electrophoresis. Okay, so in this case I'm going to take a agarose gel. So this is a agarose gel, and of course it has some thickness. So I have taken agarose gel, right? And on this agarose gel we will be seeing some small pits. So these are called wells. So these are wells. So now I told you that I have taken that purified DNA, okay, into a test tube, and then to this I have added the restriction endonuclease. So this is the DNA. Now what happened is it has cut the DNA into fragments. Now this one, that fragmented or the fragments of DNA, after treating with restriction endonucleases, I'm going to put that into these wells. So here I'm going to pour that one into these wells, right? So say that that's so of course this all just looks like a liquid. Okay, in that there will be fragments, right? And now we know that DNA is negatively charged. So this fragmented DNA will be loaded at the cathode side. So will be loaded at the, okay, cathode side, right? Because that side will be the negatively charged side here. Okay, and now this is a point. So it is connected to, okay, so the cathode, and here this side it will be connected to the anode side. So which will be of the positive side. Okay, now I'm going to supply a small voltage, right? So this electrophoresis is a small machine that will be supplying a small voltage. Okay, and the side on which I'm loading these DNA fragments will be the negative side, and that's the cathode, and the opposite side will be the anode side here. Now when I apply the voltage, what happens is that the fragments of the DNA will be moving towards this positive charge. So they will move towards this, okay, positive charge. And the smaller the fragment, so I hope you remember while discussing the DNA fingerprinting in molecular basis of inheritance, there I told you the smaller the fragment, the farther it moves. Okay, the smaller fragments will come almost near to this anode, whereas the longer fragments, they will not be able to move through the porous structure of this agarose gel. So this agarose gel will be having pores, okay, through which these DNA molecules will be moving. If the DNA molecule is lengthier, so or longer, it finds difficult to move through this agarose gel. And if it is smaller, okay, it moves very quickly and it comes towards. We know that as the DNA is negatively charged, it gets attracted towards the, okay, the positive. So here, keep in mind that I'm not separating DNA based on the charge. So I'm separating DNA based on only the size. Okay, so the DNA gets, separated, but we will be not able to see once I finish this gel electrophoresis. We will not see, of course, anything, so that the fragments have been separated. It will be not visible to, of course, to see what the DNA fragments, so how they have separated and all. Okay, of course, we'll not see that DNA molecules with our naked eye. That is not possible. Okay, uh, the point is that you will get some bands. That's how we have seen in the DNA fingerprinting, isn't it? So here actually there is a band formation that is not visible to us, and each band will contain those DNA fragments. Now to make the bands visible, I need to treat this with ethidium bromide. So once I finish this gel electrophoresis, okay, so this agarose gel is treated with the ethidium bromide, and after treating with ethidium bromide, okay, later it is exposed to UV radiation, exposed to UV radiation. So after treating with ethidium bromide and exposing it to UV radiation, I will be now able to see the fragments. I mean, able to see the bands. So here you can see the bands of various sizes. So something like this I'll be seeing. Okay, so you will be seeing something like this, right? So these are the bands, okay, which will be appearing in bright orange color. So all these will appear in bright orange color bands. So when they appear bright orange color bands, okay, after I perform the electrophoresis, okay, so by supplying a small voltage, okay, the DNA fragments will separate basing on the size, and then later I'm going to apply it on this with the ethidium bromide, and then later I'm going to expose it to ultraviolet radiation. Then I'll be able to see something, the bright orange colored bands. Okay, so now these bands will be okay, cut out from this agarose gel. So removing that, the bands from this agarose gel is a process called elution, okay. So that the separation of bands, or cutting out cutting out, okay, cutting out of bands from agarose gel is what we call gel electrophore. I'm sorry, called elution, right? So these bands, once they are formed, okay, we will be applying ethidium bromide and exposing it to UV radiation, and then once we are able to see these bands in bright orange I'm sorry, bright orange color bands. Okay, we will be removing that bands from the agarose gel in a process called, okay, elution. So after that, we will be identifying the gene of interest from those DNA fragments, and those will be used to construct our DNAs, right? So that's about the separation and isolation of DNA fragments. Then next, we'll see in detail about the cloning vectors. So here to deliver that piece of gene of interest into the host, we need to use these cloning vectors. So these cloning vectors are vehicles for delivering the gene of interest. So these cloning vectors. Okay, so these cloning vectors, especially I'm going to discuss about the plasmids. Already I have told you what these plasmids here. Okay, and the plasmids, and also something called the bacteriophages, right? So the plasmids have ability to replicate by themselves, and also the bacteriophages. I hope you remember that these bacteriophages are nothing but phages. Okay, I mean viruses. So they are viruses, and both these plasmids and bacteriophages have ability to replicate. So they can replicate, right, by themselves. Okay, uh, in the host cells, like for example the bacterial cells. Okay, and they're independent, so they don't depend on the chromosomal part of that bacterial cell or bacterial DNA. And these bacteriophages have high copy number. So what we mean by this high copy number means they can divide and can form many copies within the bacterial cell. So if it is the case, then we call it a high copy number. So these bacteriophages will have the high copy number, and the plasmids will be of only few numbers or few copies per cell. So usually they may be one or two copies per cell. Okay, of course, when we come with these plasmids, so we can find these plasmids with two types, of course, so depending on their copy number. So here we can say the plasmids with high copy number and plasmids with the low copy number, right? So plasmids with the high copy number and plasmids with the low copy number. And this means usually the high copy number that may be having 10 to 100 copies, okay, within the bacterial cell. I hope you know, in case of this bacterium, so these are plasmids and this is the bacterial genome. So here, as the plasmid has ability to replicate, so independent of the bacterial genome, it doesn't listen to this bacterial genome, or it is controlled by the bacterial genome. It is independent. So it has its own independent functions. It has its own origin of replication. So hence it will be able to replicate by itself, independent of the bacterial genome. So say suppose here I have taken the E. coli cells, and these are the plasmids. Okay, and they are able to replicate, and you can see here they are present in 3 numbers. And here if I say the plasmids which range from this 10 to 100 copies, then I can say they are, okay, high copy number plasmids. And coming to the low copy number, it may be only 1 to 4 copies per cell. So they will be, so I'll say copies per cell. and 1 to 4 copies per cell here. Okay, so high copy number and low. In this case, you can see there are around only 3 plasmids. It means that this is a low copy number plasmid. It's a low copy number plasmid here. Okay, so that's about these plasmids. Of course, in the textbook they have mentioned some around 15 to 100 copies per cell. So that may range from 1 to 2 copies per cell. So it means that here this is what they are talking about, low copy number, 1 to 2 copies per cell. So that is low copy number plasmids, or maybe from 15 to 100 copies. So if it is the case, then it is the high copy number, right? So I'll give that values here, 15 to 100 copies per host cell, right? And it will be 1 to 2 copies per host cell. Okay, so this is the values which I have taken from the textbook. Okay, so here generally I have mentioned, okay, so high copy number that may range from 10 to 100 copies per cell, and low copy number that may range from 1 to copies per cell. So this is I'll just mention the textbook values. Of course, these are the values that you need to keep in mind if anywhere in the examination they ask. Okay, so you need to say 15 to 100 copies per cell. That means the plasmid is a high copy number plasmid, and it is 1 to 2 copies per host cell, then it will be the low copy number plasmids, right? Now, if I'm going to link that gene of interest either with the bacteriophages or with the plasmid. So if I say this is the plasmid which I have taken, and to this I'm going to link this the gene of interest. Okay, so now this piece of DNA that I have linked with the plasmid will also multiply, basing on the copy number. Say suppose this plasmid is able to copy 100, okay, times, or means it can multiply 100 times, then along with that this linked DNA, right, or what I say the gene of interest will also, so say this is the gene of interest, so this gene of interest will also get, okay, copied along with the plasmid. So I can make the copies of gene of interest equal to the copy number of the plasmid. So if the copy number of the plasmid is 100, then whatever the gene that I have linked to it will also get, okay, copied 100 times, right? So that's how we can make the copies of the gene of interest by linking it to either plasmids or bacteriophages. So usually these all these bacteriophages will be having a high copy number, but when coming to the plasmids you may find two categories, one with a high copy number and one with a low copy number. Okay, so I hope this part is clear. Now, so how we will be usually joining that gene of interest with the plasmids. So what I do is I'm going to take the plasmids in a test tube, and then I'm going to take that the gene of interest into again in the same test tube, and into this I will be using the restriction endonuclease. Okay, so this restriction endonuclease, as I told, I'm going to use this echo R1. It will find this GAA-TTC palindromic sequence, and it's going to cut the plasmids. Already I have isolated this gene of interest by using the same restriction endonuclease. So hence this gene of interest will be having, okay, so those ends like AATTC, on the both sides. Okay, so that's the sticky ends we will be having. And when I use this echo R1, so here these are the plasmids, and this is the gene of interest, or you can also say foreign DNA. Now the plasmids will be open, so they are cut and they will open, right? And later I'm going to use the enzyme DNA ligase. So when I use this DNA ligase enzyme, so what happens is it will help in taking up of course this gene of interest, and some of them will get ligated to those plasmids. But some plasmids, they simply open and then close without taking the foreign DNA or the gene of interest. Foreign DNA or gene of interest. Now, those plasmids that have taken that foreign DNA, I call them, okay, recombinant DNAs. So they are called the recombinant, or I can say simply recombinates, right? And the one which did not take it, they are called non-recombinates. They are called non-recombinates. So the one who have taken that foreign DNA, they are recombinates or recombinant DNA, and these are the one who did not take. So all it is, there is no rule that all the plasmids need to take, okay, that foreign DNA. So when I add the restriction endonuclease, they are going to cut the plasmids, okay, and open them, and then okay, after applying the DNA ligase, okay, this foreign DNA will get ligated to the plasmids, but not all. So the one who have taken, they are recombinates, and the one who did not take, they are all called non-recombinates. So nowadays, we are engineering those vectors to see that which plasmid has taken the gene of interest and which one did not take it. Okay, right. So here we need to identify the recombinants from non-recombinants. We don't want non-recombinants, so what we want is only recombinants. So how we can identify those recombinants from non-recombinants, that we will shortly will come to know. I'll discuss that one shortly, right? uh, so, and I told you that these plasmids, see, because we have come to know every detail of the DNA molecules, now we have that ability to manipulate it however we want. So you can remove the nitrogen bases, you can put it, and even we have the technology to synthesize your own DNA molecules with your own sequence of nitrogen bases. So however you want. So what I'm going to do, the plasmids which I obtain it from the bacteria, okay, so I told you these plasmids, some of the bacteria will be having these plasmids. So I'm going to take that plasmids and I will be modifying them so that suit my needs. Okay, that's now. Let's see how they have modified that plasmids and then made these plasmids. And of course, to use a plasmid for this or for the purpose as a cloning vector, it should have some properties. So what that properties, the cloning vector should have something called the oricide. It should have oricide, and it also should have something called a selectable marker. So this is the one which actually helps in identifying the recombinant from non-recombinant. I told you just now that when I use this foreign DNA to ligate with the plasmid, some of them will take and some will not take it. So I need to know that which one has taken it and which one has not taken it. Okay, so I need to separate them. So for that I will be using the selectable marker which will be present within the plasmid, and I should have something called the cloning sites, means the location where I'm going to ligate that foreign DNA or that gene of interest into the plasmid. So that site we call it cloning site. So let's see the first one. So these are the properties of the cloning vector. It should have these properties to use as a cloning vector. Now first, the ORI site, we know already it's the origin of replication. So usually plasmids have one origin of replication, okay, and that is compulsory. Without this origin of replication, the plasmid will not be able to replicate by itself. If you remove this ORI site, the plasmid will never replicate. Okay, for the replication process to take place, the enzymes need to identify this site and start the process of DNA replication that we have already studied in the molecular basis of inheritance. Okay, and then this site is also responsible for controlling the number, okay, or controlling the copy number of linked DNAs. So here ORI site is also responsible for copy number controlling the copy number of linked DNA, controlling the copy number of linked DNA. Of course, how many times the plasmid will divide and form copies, it all depends on this ORI site. And when you link the foreign DNA with this ORI site, so I told you that if a plasmid is able to make its own copies of some 10, then the linked DNA along with it will also become 10 in number. Okay, means 10 copies it will form. So that's the ORI site here. Now if you want to make many copies, means whatever that linked DNA, you want many copies of that linked DNA, then you need to link that with a plasmid that supports a high copy number, right? So that's about the ORI site. And now coming to this selectable marker. So these selectable markers are usually the antibiotic resistant genes. So these selectable markers are antibiotic resistant genes. So here I'm going to take one example of this plasmid. It's called PBR322. So PBR322. So here also there is a method of giving names to these plasmids. Okay. So in this case, this P always stands for plasmid. It always stands for the plasmid, whereas the BR, it may change, it all depends on, okay, so in this case this BR is the two scientists who actually constructed this plasmid. So that B actually stands for Bolivar, one of the scientists, okay, and R stands for Rodriguez. So these are the two scientists who constructed C. Okay, so that's why they have mentioned it the BR, which stands for Bolivar and Rodriguez. And this 322 will be a numerical designation. Right. So this is how we gave the names to those plasmids. Of course, like for example, we'll see one more like PUC 19. So in this case, again, the P stands for plasmid, but this UC actually stands for university. So U stands for university and C stands for California. So University of California. So like this, it will change. I'm not going to discuss about this PUC 19. We'll see, especially with PBR 322, right, plasmid. So in this PBR 322 plasmid, we will see two antibiotic resistant genes. So there are two antibiotic resistant genes. So this is the region that codes for antibiotic resistant genes. Okay, so this is another region, and this is the one that's TET R, so which stands for tetracycline resistance. So this TET R, which means tetracycline resistant, means so that gene will produce kind of proteins which will counteract the tetracycline. So tetracycline is antibiotic. Tetracycline is antibiotic, and in this case this is the AMPR, so which stands for so ampicillin resistance. So this AMPR stands for ampicillin resistance. So ampicillin resistant, so resistant to ampicillin antibiotic. Okay, so these two will be the selectable markers in this case of PBR 322. Of course you can use other antibiotic resistance, okay, to like for example kanamycin resistance, okay, chloramphenicol resistance, and so you can use it. But in this case of PBR 322, the scientists have made in such a way that it has only two selectable markers. Now how they are going to act as a selectable marker? Say that this tetracycline resistance has certain restriction sites. So restriction sites, which means the locations where the restrictions enzymes, restriction endonucleases are going to cut. Okay, when we see this tetracycline resistant, in this case there are actually two restriction sites like BAM-H1. So this is a restriction endonuclease. If I use this BAM-H1, it's going to cut at this point. And there is one more called SAL1, that is another restriction endonuclease, and later I will give you what's this BAM H1 and SAL, what's the name of this bacteria. I hope already you know that how we will be giving names to restriction endonucleases. So in this case the BAM H1 will be from Bacillus, okay, the amylo liquefacients, right? So this is Bacillus amyloliquefacients. Okay, from that we have obtained this restriction endonuclease. And in case of SAL1, so that is obtained from Streptomyces albus. I'll give that names and all later. So these are the two recognition sites. If I use this BAM H1, what it will cut at this point, and now when I link a foreign DNA. So here is the foreign DNA, and if I'm going to link this foreign DNA at this BAM H1 site, so if I use this restriction endonuclease BAM H1, so it's going to cut open the plasmid, and now I have introduced this one here, okay, the gene of interest. Now, because of inserting that foreign DNA into this tetracycline gene, what happens is the tetracycline gene becomes non-functional. The tetracycline gene becomes non-functional. Okay, so insertion of foreign DNA. So insertion of foreign DNA into Okay, TET-R gene makes TET-R gene non-functional. Means, okay, so it makes the TET-R gene to become non-functional. It will be no of use. It will be now. This will actually help me to identify the classmates that have taken that foreign DNA and the one which did not take it. So of course, in this case, when it has taken, it's called the recombinant, and the one if it doesn't take, then it becomes the non-recombinant. So later I will discuss in detail that how we will be able to identify basing on this which one is a recombinant and which one is not a non-recombinant. So at present, so just we'll see the structure of this cloning vector. Okay, then coming to ampicillin resistance, in this we have again two more recognition sites. So one, it is PVU. So at this point we have PVU1. Okay, and below that we have one more, it's a PST1. And then we will be seeing here